What happens when part of the endomembrane system does not function properly? Let's get into it. The endomembrane system is a network of interconnected organelles that work together to synthesize, modify, package, and transport proteins and lipids. It includes the endoplasmic reticulum, which is comprised of the rough ER and the smooth ER. Rough ER is studded with ribosomes and is the site of protein synthesis and initial post-translational modifications. The post-translational modifications that occur in the rough ER include protein folding, disulfide bond formation, glycosylation, which is the addition of carbohydrate chains, and quality control, ensuring that proteins are properly structured and functional before being transported to the Golgi apparatus. Smooth ER lacks ribosomes and is involved in lipid metabolism, detoxification of drugs and toxins, and storage of calcium ions. The Golgi apparatus is a series of stacked membrane-bound sacs that further modify proteins and lipids received in the ER. It functions in sorting, packaging, and directing molecules to their appropriate destinations, including secretion via vesicles. Lysosomes are membrane-bound organelles containing hydrolytic enzymes that digest worn-out cellular components, pathogens, and macromolecules. Lysosomes are critical for cellular waste management and recycling processes. Transport vesicles are small membrane-bound structures that move molecules between organelles in the endomembrane system or to the plasma membrane for exocytosis. So now, what happens when one or more of these structures don't function properly? Cystic fibrosis is caused by a mutation in the CFTR gene that leads to improper protein folding in the rough ER. The misfolded CFTR protein is retained by the rough ER and prevented from reaching the Golgi apparatus. Normally, properly folded proteins are transported from the rough ER to the Golgi for further processing and trafficking. However, in cystic fibrosis, the ER's quality control system recognizes the misfolded CFTR protein as defective and targets it for degradation. This prevents the CFTR protein from reaching the plasma membrane, leading to defective chloride ion transport and the buildup of thick mucus in the lungs and digestive system. Thick, sticky mucus buildup results in chronic respiratory infections, persistent coughing, wheezing, and difficulty breathing. It also interferes with the digestion, leading to malabsorption, poor weight gain, greasy stools, and in some cases, diabetes due to pancreatic dysfunction. Deficiencies in lysosomal enzymes, such as in Tay-Sachs disease or Gaucher disease, lead to the buildup of toxic substances, causing progressive cellular dysfunction. Tay-Sachs disease is a genetic disorder caused by a deficiency of the enzyme hexosaminidase A, leading to the accumulation of GM2 gangliosides in neurons, resulting in progressive neurodegeneration, muscle weakness, vision loss, and early childhood death. Gaucher disease, caused by a deficiency of glucocerebrosidase, leads to the buildup of glucocerebrosides in macrophages, causing symptoms like enlarged liver and spleen, bone pain, anemia, and neurological complications in severe forms. Let's turn our attention to peroxisomes, which are small membrane-bound organelles that contain oxidative enzymes. Their primary roles include detoxification, so the breakdown of hydrogen peroxide, a byproduct of metabolism, using the enzyme catalase to prevent oxidative damage, metabolism of alcohol and other harmful substances in liver cells, fatty acid oxidation. Peroxisomes are involved in the beta oxidation of very long-chain fatty acids, which mitochondria cannot process efficiently. They are also involved in the biosynthesis of key molecules. For example, the synthesis of plasmalogens, which are essential phospholipids in myelin sheaths, critical for proper nerve function. An example of a peroxisomal disorder is Zellweger syndrome. This is caused by defective peroxisomal assembly, leading to the accumulation of very long-chain fatty acids and neurological dysfunction. In summary, the endomembrane system plays a central role in the synthesis, modification, and transport of proteins and lipids, while peroxisomes handle detoxification and lipid metabolism. Dysfunction in these organelles can lead to severe metabolic and neurodegenerative diseases, highlighting their importance in human physiology.